right, good day, everyone. I am doing part two of this week's and next week's lecture. Uh, the first part I had talked about host microbe relationships and did some review of the relationships, the symbiotic relationships. Also talked about the microbiomes and how we acquire our microbiomes, and also talked about some of the microbiome's importance to us, and a little bit about the gut brain axis and how it helps in maintaining homeostasis and how it even affects our mood. I also talked about dysbiosis and eubiosis and some of the characteristics of what occurs during that either stage. And then finally, there was some new articles or research that's being done on the human gut microbiome, as far as multiple sclerosis and fibromyalgia, and also even autism. So tonight, I'm going to talk about further about infection and disease. This is the meat of this week's lecture. And again, I want to emphasize that not all, not even most, but a few, only a few of the microbes cause human diseases. And I mean, it's like 1/100%. Very small amount of known microbes actually cause us disease. Now, interesting, the stages of disease also correspond to the bacterial growth in a population. We have the incubation period, and at that point, we've just been newly infected. A person has just been newly infected. So the microbes haven't really established within that host yet. That's the incubation period. And as the number of pathogens increase, multiply and increase, so do the symptoms and signs of illness in the humans. At one point, probably at the log phase of that bacterial population, we will have corn sensing taking place, okay? And if you remember that, that's when the cells start signaling to each other. What are they signaling for? They start producing toxins, expressing those genes and producing virulence factors, toxins. And at that point, when this is starting to be produced by the bacteria, our immune system is in full activation. So that is a time where we're in period of illness, where the signs and the symptoms are at their highest expression. So anyhow, what happens? Our immune system starts kicking in, and in the infection, normally we have a period of decline. And it has to do with the population starts to decrease because our immune systems are dealing effectively with that infection. And if we've got a good immune system, at some point, hopefully not too long, sterilizing immunity is achieved. In other words, the pathogen has been conquered. At that point, we have a period of convalescence where we are healing, our cells are healing, and we will return to a healthy condition. Now, in the next slide, how are these 1%ers, these pathogens, how do they cause human diseases? Well, they've got pretty much three attributes. First of all, they have short generation times. So the cell numbers can increase rapidly. Also, these bacteria will express genes for virulence factors. So these are factors that may help them in infection, but it will hinder or harm us during that time. And then finally, our response to the virulence factors will transmit that pathogen to new hosts. So at some point, our immune system kicks in and maybe conquers or gets rid of that pathogen. But also at some point, these pathogens will run out of nutrients and then they need to move on to new hosts. And they'll do that by pretty much exiting the body in a similar way or through the same system in which they entered. So down here at the bottom, what we have is a scheme of an infection. Okay, each of these boxes is showing different stages in an infection, what the bacteria does in an infection. So first off, they have to enter the host. And bacteria can enter, depending on the type of bacteria, they are only infectious, will only cause a disease if they're able to enter in a specific way. So they'll enter either a mucous membrane, through the respiratory tract, the GI tract, the genital urinary tract, the eye, the skin, or even the blood. So once they've entered, they will want to penetrate, invade, or avoid the host defenses. I want to mention here too, when a bacteria or pathogen enters, two factors come into play as to aiding the infection, and that is you have to have a number of invading microbes. Okay, you can't just usually have one. The higher the number, more times than not, the higher the number, the more likelihood of an infectious disease. Also, many of these microbes have to be able to adhere, to stick to the host cell or to the host tissue. So once they get inside the host, then they may start displaying their virulence factors. And some of the virulence factors don't necessarily cause damage, but they do aid the bacteria in infecting. Okay, and so one of those is a capsule. Also, they may have enzymes that will help penetrate the host cell or tissue. Capsules will evade the host defenses. So anyhow, and once that is, entry has been established, entry has penetration or evasion from the host defenses has happened, then we get into the damage to the host cells. And we've got here listed several things that damage the host cells, because remember in the earlier video, that's what a pathogen does. Okay, what makes pathogen a pathogen is it causes damage to the host. So these are some of the things here in that purple box that are damaging to the host cells. The ones that we'll talk about are the toxins today, and maybe a couple of the other things that cause direct damage. So once they've come in, they've entered, they established, they've produced exotoxins, multiplied. What they're going to do next is, at some point, if not completely overtaken by the host immune system, want to leave the host, and they will leave through a portal of exit. And in many situations, or generally, they will leave the same way in which they entered the host. So in other words, if a host has acquired that infectious disease through the respiratory tract, say it's a cold or the flu, it will then at some point leave through a cough through the respiratory tract. And at that point, then that pathogen will be capable of transmitting to another host. So anyhow, some of the bacterial virulence factors, one in particular, a well-known one, is your capsule. And if you see in that picture there, we've got some encapsulated bacteria. They actually are diplococci. And then you've got a non-encapsulated bacteria. And what these capsules do, remember they're like a sugar coating around some bacteria. They will basically not allow a phagocytic cell a cell from the host immune system to attach to the bacterial cell, because that bacterial cell has that capsule coating, and that will protect it or inhibit the phagocytic host cell from attaching. And if it can't attach, it won't be able to digest that bacterial cell. Also, some of the host cells will have antibodies. The antibodies too are unable to attach to that bacterial cell. In this case, you'll see in the picture, the second picture, the bacterial cell is releasing protease, and I will talk about that in a little bit. That is an enzyme that will break down certain proteins. In this case, this specific protease is breaking down the antibody. Okay. Another virulence factor is pili. Now, if you remember, the pili are used for attachment. I'm not talking about the conjugation pili. That is used for something other than attachment. But I am talking about the pili that attach. And we've got two types, type 1 and type 4. Type 1 is the one you typically think of for attachment. They have these little sticky compounds, these adhesins, at the end of the pili that will attach to a host cell. Okay, this type 4 though of pili, yes, it attaches to a surface, but the pili is mainly used for movement. So what happens in this case is that the bacterial cell will extend its pili. It kind of grows a pili, I guess it would be, extends it, and then it will attach to a surface or to a cell, and then start contracting. As that pili starts to contract, it will pull the bacteria. And if you've got several bacteria on a cell or on the surface, it will end up pulling them closer together. And it's, again, this pili is associated with movement, a twitching and crawling type of movement. And I believe that the website down at the bottom of the slide will show you that type of movement. And it is important in infection, in infecting cells, host cells, but also this type of pili is used in the establishment of biofilms. Okay. So type 1 has adhesins and used for attachment. Type 4 is more or less attachment. In addition, though, it's used for movement, a twitching and crawling type of movement that aids it in infection. So this is a case of those two type of pili. And these two types of pili are found in certain strains of E. coli that cause hemorrhagic diarrhea. Some of them do, some of them cause just diarrhea, but some are bloody diarrhea. And they are pathogenic variants of E. coli. So we have a strain of E. coli that's in our gut that is not pathogenic. But then again, you've got some cousins of it. Some of its cousins, they are strains that have become pathogenic. And we've got two with type 1, and they're showing you numbers. If you look at the one on the far right top, type C, and then the far right bottom row, type F, have this type of pili. The attaching type of pili. And then if you see, we've got A, B, D, and E. The other 4 are pathogenic strains of E. coli that have the type 4 pili, the bundle forming, the twitching and crawling, and they form type of bundles. Again, if you look at D, that strain is pretty common in biofilm productions. Okay? You don't have to memorize all these types or strains of pathogenic E. coli. It's just to show you that in the pathogenic strains, they do have these types of pili as virulence factors. These pili genes again are motile genetic elements, big word for bits of DNA or plasmids that are spread by horizontal gene transfer. So each one of those strains basically mutated from a commensal E. coli from our gut, and they mutated basically by acquiring extra pieces of genes or extra genes that would be expressed that would express these specific types of pili. And all of a sudden that helps them in crossing the line from a commensal to a pathogenic bacterium. Then we have also secretion systems. So you've got these toxins or these virulence factors that are being made by the bacteria. How do they get into the host cell? So one of the ways they can is by channels. So the bacterial cell will have pretty much channels, protein channels, in which that virulence factor will be released and taken up by the host cell. The second type of secretion system, there's a type 3 secretion system, and this is in the bacterial cell. The protein or the virulent factor is then pretty much injected into the host cell. So these are two types of ways in which the proteins or the virulence factors are delivered to the host cells. Okay, another virulence factor are your exoenzymes. So if you look at the word enzymes, they basically are enzymes that are released through channels from the pathogenic bacteria. And what they do is they facilitate or help that bacteria in spreading. And it will do that by either breaking cells apart or even breaking tissues down so that bacterial cell can spread throughout the host. And these are proteases, okay, a type of enzyme that degrades certain proteins of the host cells, in between the cells or within a tissue. And we've got two examples here. We have hyaluronidase, which will break, as you see there, the hyaluronidase, the enzyme, will break apart the tight junction between the cells. And also collagenase, do not confuse that with collagen or with coagulase. But collagenase, as you see that word, it is an enzyme that breaks apart the tissue so that bacteria may spread. Okay, now I'm going to talk about the endotoxins and exotoxins. And you may be very familiar with hearing these, endotoxins and exotoxins. These are the poisons produced by bacterial pathogens. And there are two main groups, endotoxin, and the second group is exotoxin. Okay, the characteristic of the endotoxin is that they're usually gram-negative bacteria. Their toxin is lipid A, component of the lipopolysaccharide. So remember that LPS, that lipopolysaccharide, that's in the outer membrane of the gram-negative bacterial cell, well, guess what? That is the toxin of the endotoxin, or is an endotoxin and the toxin of the gram-negative bacteria. Usually, what effect does it have on the host? This usually will cause symptoms such as inflammation and fever. And these toxins are pretty heat stable, so heat won't degrade them. With the exotoxins, these are usually found on gram-positive bacteria and maybe some gram-negatives, but usually primarily gram-positive. And as the name says, exotoxin, these toxins are released into the environment. They are protein, composed of protein, and these guys will cause specific damage to cells. Okay, these do cause specific damage. Most of these exotoxins are sensitive to heat, but some are pretty stable in heat. And the main difference I want to put here about the composition between exotoxins and endotoxins is that with an endotoxin, those in the gram-negatives, the lipid A of the lipopolysaccharide is within the cell wall. In the exotoxin, with the gram-positives, these are released into the environment. Endotoxins are usually released when the bacterial cell dies. Okay? So getting a closer view on the gram-negative cell wall structure, again, I said that the lipopolysaccharide is the endotoxin. And here I'm just going to show you with the arrow, this is your outer membrane of the gram-negative cell wall. And you've got the LPS, lipopolysaccharide, right here. Your lipid A is the toxin part, which is embedded in the outer membrane. And if you look right here, this chain here are your polysaccharide. It's a chain of sugars. So what happens is when this is released from the bacterial cell, and especially your lipid part A, it will generate a reaction. It will activate the macrophages. It activates the protein signaling system of the immune system, complement. And it also can activate tissue factor. The macrophages may induce or result in fever. It could in hypotension. And then in some cases, complement, and you've got hypotension, may lead to edema. You may get the movement of white blood cells to this spot. And then some clotting and even DIC. So in some cases, this can be, if you get enough of these endotoxins, a serious reaction to them. All right, a little bit about exotoxins, and I think we're going to be ending here. Exotoxins, again, those are made by gram-positives mostly, and they are the guys that directly damage cells and tissues. So if you think of the word exo, think of out of. So these are the toxins that are released from the bacterial cell into the environment, and they are the ones that cause the damage. And there are three types of exotoxins. We've got the intracellular targeting toxins, or the A-B exotoxins. A second type are your membrane disrupting toxins. And finally, a third type of exotoxins are your super antigens. So the intracellular targeting toxins, your AB exotoxins, As you see, you've got two letters there, A, B. And there are two parts to this exotoxin. You've got one part, A, that is the toxin itself, and then B is the binding part of the toxin. So the B part will first of all, carrying the A, bind to the host cell or the target cell. The A part that holds the toxin is then introduced into the host cell, and that is what will produce, when they are expressed, produce the damage or damage the host cell. So we've got categories or examples of the toxins in this category, in the AB exotoxins, and cholera and tetanus, botulinum and diphtheria, all are examples of the AB exotoxins. All right, the second type of exotoxin, membrane disrupting toxin, are your streptolysins, your pneumolysins, alpha toxin, beta toxin. And what these guys do is they pretty much, they are a set of proteins that assemble on the bacterial cell membrane, and they poke holes in the cell membrane. They make the holes or pores into that host cell, which kills it. And again, these are, we've got in the column there, we've got examples of those membrane disrupting toxins. And you see that a couple of them are found in the Streptococcus pneumoniae. One's found in Staph aureus. And you've got your Pseudomonas there. That's Pseudomonas with the phospholipase C. So these will all affect the host cell membranes. Finally, we have the superantigens. And what these guys do is they pretty much stimulate an excessive activation of the immune cells, producing a lot of cytokines. And the result is, actually, it can be life-threatening. You've got fever, inflammation, and shock. Some of the examples of the superantigens are your toxic shock syndrome toxin caused by Staph aureus, and some streptococcal pyrogenic toxins. These are found in Streptococcus pyogenes. So luckily there aren't, these aren't very common, these superantigens, but they are there in nature. So anyhow, I'm gonna end here. I hope you all have a nice weekend. We are getting close to the end of the semester, and I will continue with the next week's notes next week.